what?
Bacteria.
Bacteria, and what kingdom would that be?
Monera.
Monera, good. What domain would that be?
Prokaryote.
Domain.
Prokaryote.
Bacteria, or it could be, if I'm studying the primitive bacteria, what domain?
Archaea.
Archaea, right. All bacteria are prokaryotes. All prokaryotes are bacteria. Everything else is a eukaryote. That's where that's coming from. They do call kingdom Monera kingdom prokaryote, but that's not a domain. So what's the domain then of eukaryotes? Eukaryote. Right. And some people call it eukaryota, but I use eukaryote for the domain name. Okay, if we're studying molds and fungi, what is that study called?
Mycology.
Mycology, good. And what kingdom would that be?
Fungi.
Fungi. And what domain would that be? Eukaryote. What if I'm studying phycology? What am I studying?
Algae.
Algae. And algae come from what kingdom?
Protista.
And?
Planta.
And?
Monera.
Monera. What kind of monerans are algae?
Cyanobacteria.
Cyanobacteria. Good. And that would be what domain?
Protista.
For algae. Could be either domain. Because planta is domain eukarya. Protista is domain eukarya. But cyanobacteria is domain bacteria. Or it could be archaeobacteria, depending on the type of cyanobacteria. Alrighty, and virology is the study of
Viruses.
How do we classify viruses?
Genetic material.
Right, based on the type of genetic material they have, based on the type of nucleic acid they have. And we even give them family names and genus names because we're imitating the classification scheme for living organisms. What kingdom is viruses?
None.
No kingdom, right. What domain is viruses?
None.
None. They'll probably come up with one. They'll probably imitate again, but they haven't really. Have they settled on a virus kingdom name or anything? Not as far as I know. I'm sure it's a topic of discussion amid some bunch of nerds, but until they tell me in the textbook, I'm not going to worry about it. Okay, did we miss any kind of, what's the study of helminthology?
Worms.
Worms, good. And worms are what kingdom? Animalia, good. And how about insects and arachnids? Yeah, that would be domain eukarya, and that would be kingdom Animalia. And actually, the study of insects is entomology, and the study of arachnids is, I have no idea. Arachnidology? Maybe, I don't know. Arachnophobia? I don't know. It is arachnology? I actually did some entomology for my undergraduate research project. I taught predaceous diving beetle larvae to avoid food choices. Some foods they loved and they glom right on, but if you gave them a little tadpole of a toad, they'd go like, get that thing away from me, because it has toxins in it, so the predaceous diving beetle larvae won't eat the tadpoles. Mother Nature's got all kinds of weird mechanisms going on. All right, but it's interesting that they learn. Little insects actually learn. They're not stupid. And they're so smart, in fact, so I had all these predaceous diving beetle larvae, Google them, and it's like an insect about that big, and it has these giant mandibles, and it grabs its food with these giant, like a big ice pick thing, you know. And when it's like their regular feeding time, they know what time they get fed. When it gets close to feeding time, they all start banging their mandibles against the side of the plastic containers that they're in. Their insects are really smart. It's amazing. My girlfriend came to visit me at the research station. She was all grossed out. She's a banker. She wasn't into that bug. Alrighty. still know some of your organisms, so, you know, go back through your parasite list, go back through your bacteria list, and you just did viruses, so you know your
very
major
broad.
viruses. And you'll get choices sometimes, which one is a mole, which one is a worm, which one is a insect, and which one is bacterial, and that kind of thing. So what kind of organisms are in Burgess' manual? Bacteria
Bacteria.
only.
Yeah.
So there was a question on one of your tests about which organism would you expect to find in Burgess' manual, and there was only one bacterium in the list. Alrighty, don't forget your shapes and arrangements. And you have some of that on your lab practical also. So you have cocci are round, bacilli are rod-shaped, and spirillum are spiral-shaped, and spirochetes are also spiral-shaped, perhaps a little more corkscrewy. The spirillum have flagella, the spirochetes do not. They have an axial filament inside, an endoflagellum inside instead. Then you can qualify that. You might have a streptobacillus, a chain of bacilli. There's no such thing as a staphylobacillus. You could have a streptococcus, a chain of cocci, looks like a pearl necklace. You could have a staphylococcus, a cluster of cocci. If the cluster is a regular grouping of 4, it's a tetrad. If it's a regular grouping in a box shape of 8, then it's a sarcina. Other clusters we just call staphylococci. But tetrads and sarcina are staphylococci. Then there's also diplococci. That would be little 2 together. Also, there can be a diplobacillus. Usually it means the bacillus is just getting ready to divide into 2 cells. But some bacteria of cocci remain together as diplococci. And that's the normal arrangement. Alrighty, don't forget your group A strep, the group B strep, and then you have your enterococcus, which was group B strep, but now it's enterococcus. So you need to know your different strep groups, so pull up that sheet of information in the lecture material, it's still there. Alrighty, and don't forget your stains. We did simple stains of 2 kinds. We did a direct stain, which stains your organism directly. And the 4 direct stains we used, we had purple, we had blue, methylene blue, purple's crystal violet, and we had brilliant green, and we had saffron, which is red. Then we did a negative stain, in which the cells are clear and the background is stained, and we used nigrosin, which is black, and we also used Congo red. Eosin is another negative stain, which is like a purpley red, dark red. And we also did differential stains. The gram stain is a differential stain. Gram-positive bacteria come out purple from the crystal violet. Gram-negative bacteria come out pale red from the saffron. The other differential stain was the acid-fast stain. The TB commune stain was the type of acid-fast stain we did. And your bacteria come out either pink because they're acid-fast, which means they have mycolic acid waxes in the wall, or they come out green, which is because they don't have the waxes in their wall. And we did some special stains, but you don't have to worry about the stain procedure. You just have to know the structures. So let's do a little bit about bacterial structures. Bacteria have a capsule. Capsule helps them stick to host tissues or stick to environmental surfaces, and also helps them hide from phagocytes and resist being caught by phagocytes, so it resists phagocytosis. If they have capsules, sometimes they can join their capsules together and form a biofilm, and a biofilm makes them more antibiotic resistant because the antibiotic can't penetrate the biofilm. Remember, Streptococcus mutans is normal flora in your mouth. Streptococcus sanguinis or sanguis is normal flora in your mouth, and they have a tendency to form those biofilms. And that's what plaque is in your mouth. It's a biofilm that you let go. You can brush your teeth for a long time. And then some other bacteria, the anaerobic ones, can grow behind the biofilm of the streptococcus and form periodontal disease. Alrighty, don't forget about your capsule. So let's do endospore. Endospore, you should be able to recognize when you see bacteria with endospores in it. There's only two genera that can produce endospores.
Clostridium and Bacillus.
Good. And we did lots of different species. We did Clostridium perfringens, which is gangrene, Clostridium tetani, tetanus, Clostridium botulinum, botulism, and Clostridium difficile, C. diff, diarrhea. And we also talked about Bacillus anthracis. We talked about Bacillus subtilis when we used it in lab. And there's lots of different species of Bacillus. But Bacillus anthracis is probably the easiest one to remember. And then another structure we looked at was flagella, which obviously is for motility. The spirochetes that have the axial filament, that also helps them with motility, but also helps them actually corkscrew in, penetrate tissues. And Treponema pallidum, which is the spirochete, that one can even penetrate bone. They can actually dig up bones of people who died like hundreds of years ago and see scars in the bones where the treponema burrowed into the bone. And they knew that that person had syphilis when they died. It doesn't mean they died of syphilis, but they, in the olden days, you lived with syphilis throughout your lifetime. In fact, we talked about, did I mention Al Capone was in a asylum for syphilitic patients, which I had in those days, because if you lived with syphilis your whole life, eventually it was gonna